Hello everyone. This video covers mostly Chapter 8 in Essential Biochemistry. So in this chapter we talk about lipids and membranes, and you can see from this picture right here that membranes are fluid, and the motion here is meant to indicate that. So let's start with how we classify lipids, which is not easy to do, as you can look at this diagram right here. So we can classify lipids as having fatty acids as part of their structures or not. So let's note that lipids don't form polymers the way we saw with amino acids forming proteins or nucleotides forming nucleic acids, but they do form aggregates. So we can have lipids that have fatty acids as part of their structure or not. Eicosanoids are signaling molecules. Triacylglycerols are oxidized for energy. And sphingolipids and glycerophospholipids are found in membranes. So in this class, we will mostly focus on the lipids that we see in membranes and the triacylglycerols that are oxidized for energy. So if we want to talk about the kinds of lipids that have fatty acids as part of their structures, we need to talk about what a fatty acid is. So basically a fatty acid is simply a long chain carboxylic acid. So we have the carboxylate group right here. The pKa is about 4.5, so that means it's going to be ionized at physiological pH. And if you look at this chain right here, this shows 12 carbons. So chemists would start the labeling at carbon number 1 to the end. If you study nutrition, the nutritionist will start in from the next carbon in and call that carbon alpha. And here they show alpha, beta, gamma, delta, epsilon, and then they stop, because it doesn't matter where you are in the Greek alphabet, the last carbon is the omega carbon. So this entire thing is a fatty acid. If we're looking at just this portion right here, that's the fatty acyl group, and this portion right here is the hydrocarbon tail. So this particular picture right here shows a saturated fatty acid. There are no carbon-carbon double bonds. We can have monounsaturated fatty acids that have one cis carbon-carbon double bond. Polyunsaturated fatty acids have 2 or more cis carbon-carbon double bonds, and trans fatty acids have 1 or more trans carbon-carbon double bonds. So here are some examples. So this shows the acidic form. So here the carboxylic acid group is shown protonated. So that's why the names are stearic acid, oleic acid, linoleic acid, and alpha-linolenic acid. If we pull the proton off, we would call it stearate, oleate, linoleate, and alpha-linolenate. So what I want to point out here is so here the carbons are labeled the way a chemist would, but if we would label them the way a nutritionist would, where you come down here to the methyl end and look at the omega carbon, that we find that stearic acid, or stearate, if it doesn't have its proton, is completely saturated, no carbon-carbon double bonds. Oleic acid or oleate is what we call an omega-9 fatty acid. So we have 1, 2, 3, 4, 5, 6, 7, 8, 9. The first double bond is encountered at the number 9 position, counting from the omega end. Omega-9 fatty acids are not essential. Our bodies are able to make them. But linoleic acid and alpha-linolenic acid are essential. So these are omega-6s, 1, 2, 3, 4, 5, 6. There are multiple double bonds. The first one is encountered at position number 6, counting at the omega end. And linolenic acid has multiple double bonds, but again, counting from the omega end, 1, 2, 3. The first carbon-carbon double bond is encountered at the omega-3 position. So omega-6 and omega-3 fatty acids are essential, which means we have to have them in our diets. Our bodies cannot make them. So we want to look at the melting points of fatty acids because that gives us an idea of how they got the names that they do. So if we would look at just simply saturated fatty acids, we can see that as the number of carbons goes up, the melting point goes up. If we would just consider the same number of carbons but looking at the number of double bonds, we see that as the number of double bonds goes up, the melting point goes way down. So these diagrams right here can explain why. So these are our ball and stick models right here. So a saturated fatty acid has no double bonds. We have our monounsaturated with one double bond, or polyunsaturated with many double bonds. And the way that these are generally synthesized, the double bonds are in the cis configuration. That puts a big bend in the shape of the molecule, and they can't pack together very well. You can imagine if you have a saturated fatty acid that they pack together with their neighbors very well. So if you can't pack together very well, then you don't have that many London dispersion forces. So if you have a great many London dispersion forces the way you would with a saturated fatty acid, then the melting point is going to be a lot higher. It's going to take a lot more heat to disrupt them. But these monounsaturated and polyunsaturated fatty acids with that cis double bond and the big bend that it produces, the fatty acids don't pack together very well. There aren't as many London dispersion forces between them, so it doesn't take that much heat to disrupt them, so the melting points are lower. So now that we have some idea of what fatty acids look like, the lipids that have fatty acids as part of their structures are triacylglycerols. Our book calls these glycerophospholipids, generally just the term phospholipids is used, and sphingolipids. I am going to show you the structure of steroids just so we're familiar with it, but steroids generally don't have fatty acids as part of their structure. So first, let's talk about triacylglycerols. So the dietary fats and oils that we love to eat are triacylglycerols. They are neutral and nonpolar. They are stored as fat globules in adipocytes. So adipocytes are fat cells that compose the adipose or the fat tissue. They yield a great deal of free energy when you oxidize them, as we will find out in a later chapter. And if you looked at the structure of a fat globule, the fat itself occupies nearly the entire cell. The other kinds of organelles that we're accustomed to seeing in the cell are kind of squashed into a smaller space. We do know from our experience that oil and water don't mix, so our triacylglycerols are completely nonpolar. They don't mix with water at all. So let's look and see how we can build a triacylglycerol. You might remember from organic chemistry that if you have an alcohol and a carboxylic acid, that they react to form an ester. So if I take off this OH from the alcohol, and if I look at that carboxylic acid, but if I just take off the hydrogen, I can form an ester functional group. Biochemists refer to that as an acyl functional group. And then this is a condensation reaction, so we have water as our product. So again, we take off this OH and that hydrogen to get water, and we've formed an ester or an acyl functional group. So the way we form a triacylglycerol is we start with glycerol as our backbone. And again, you might remember from organic chemistry that glycerol has 3 carbons and 3 OH groups. That means that we can have 3 fatty acids forming that reaction with the carboxylate group and the OH group to form these ester or these acyl groups. So that's why we call these triacylglycerols, because glycerol, shown here in black, is the backbone. We have 3 fatty acids that react to form an ester, so we have 3 acyl groups. So we have a triacylglycerol. So we have 3 fatty acids that react to form these ester or these acyl linkages. So you might notice if you look at carbon number 2, this is a different drawing right here where the stereochemistry is shown, that carbon number 2 is chiral. So even if you had all 3 of these fatty acids as identical, this is not a superimposable isomer. The carbon number 2 is chiral, so there's a chirality associated to these molecules. So I showed you the fatty acids with their different melting points. So the melting point of a triacylglycerol depends on what fatty acids make them up. So if we have a high melting point triacylglycerol that contains mainly saturated fatty acids, that's what we call a fat. It's a solid at room temperature. Whereas if we have triacylglycerols that are oils and they have mainly unsaturated fatty acids, remember those have lower melting points, so they will be liquids at room temperature. So the fats and the oils that we love to eat are all triacylglycerols, but they have different fatty acids that make them up. So dietary sources are actually not a single type of triacylglycerol. They're usually mixtures. If we look at something like olive oil, you can see that olive oil is mainly consists of carbons 16 and 18 fatty acid chains, and mainly they're unsaturated. We have a small amount of smaller sized saturated fatty acids and some C16 and C18 saturated fatty acids. So you can contrast that with butter and beef fat, where you can see this dark blue indicates that butter, which is sort of a soft solid, and beef fat, which is a hard solid, that the less liquidy you get and the more solid you get at room temperature, that indicates a high degree of saturated fatty acids. We're interested in that because for a variety of reasons, some that are understood and some that are not, diets that are high in saturated fats are correlated with heart disease. So that's why it's preferable to have a diet that's in which you use things like olive oil rather than butter or beef fat. So you might have heard of trans fats. So you can take an oil and you can carry out a partial hydrogenation where you convert some of the carbon-carbon bonds to carbon single bonds. That's going to increase the melting point. But it turns out that trans double bonds are more stable, so during that process, some of the cis carbon-carbon double bonds will be converted to trans. So this is a birthday card that our former secretary gave to me on my birthday a few years ago, and she says, "I believe that trans fat is just the scientific term for extra tasty," which is true. That's why it's been difficult to get trans fats out of our food sources, because they do taste really good. They have this nice mouthfeel. So things like creamy foods, donuts, and creamy things like frosting, they have lots of trans fatty acids. So I have another cartoon that a relative sent me that said they'll be screaming a lot louder if they knew it was trans fat. So I wanted to show you where you can find trans fat. So fresh French fries, breaded fish burger, anything that's breaded, pizza, tortilla chips, donuts, muffins, chocolate bars, a lot of them might have trans fatty acids in them. The way that you tell is that you can look on the label, and if the label says partially hydrogenated vegetable oil, that means the food has trans fatty acids in it. The FDA actually allows food manufacturers, if there's less than half a gram, to say there's zero. So for example, this chocolate bar right here that had 0.2 grams of trans fat, the label actually would say 0. They're allowed to do that. So look on the label. If it says partially hydrogenated vegetable oil, it has trans fat. Okay, so those are the triacylglycerols that can compose the fats and the oils of our diets. So let's transition to phospholipids. These are the major components of cell membranes. So unlike the triacylglycerols, they are amphipathic. So we build these by starting out with glycerol, but we are going to take carbon number 3 and we're going to attach a phosphate group. So our backbone is glycerol 3-phosphate. That leaves us 2 OH groups that we can use to esterify fatty acids. The carbon number 1 usually has a saturated fatty acid, and carbon number 2 is usually unsaturated. So that means you get something that looks like this. So here's our glycerol shown in black. Here's our fatty acid number 1, fatty acid number 2. There's the phosphate. And in green, we can also use our phosphate to as an attachment point for a head group. So the types of head groups you can have that are shown by X are things like ethanolamine, choline, or serine, and you can see that these are polar charged groups. So we would just call these phosphatidylethylamine, phosphatidylcholine. You might see that's also called lecithin. You might see that on your food packages, or phosphatidylserine. So here the phospholipids, because they have the same basic structure, are named for their head groups. So here's the structure of a phosphatidylcholine. So again, let's see how it was built. In black, that's the glycerol. Carbon number 3 has the phosphate, shown in blue. And then this is choline right here, shown in green. And there's carbon number 1 with an 18 carbon fatty acid, completely saturated, so it's stearate. And carbon number 2 has oleate, so 1 double bond. So we can, if we wanted to name it precisely where we identify the fatty acids, we could call it 1 stearoyl, 2 oleoyl, 3 phosphatidylcholine. And the structure looks like this. The CPK coloring isn't strictly observed here, where the artist here has used green instead of black, which we typically use for carbon. But you can see the amphipathic nature if you sort of substitute in your mind the green for black, where we have a polar head group and 2 nonpolar tails. Again, this is the saturated fatty acid, and this is the unsaturated fatty acid with the big bend in it. We can draw it like this, where we have our glycerol. There's carbon number 1 that is saturated. There's carbon number 2 with the big bend and the double bond, and the phosphate and the choline. So again, this portion of the molecule is polar or hydrophilic. We can call that the head group, and we have 2 nonpolar tails. So we can draw it schematically like this. So the artist here drew this polar head group in blue to represent water. So this is hydrophilic or water-loving. These nonpolar tails drawn in sort of a yellow, oily kind of color are hydrophobic or water-fearing. So these molecules are amphipathic. They have hydrophilic and hydrophobic domains in the same molecule. So here are some structures of some phospholipids, along with sphingomyelin, which I haven't talked about yet. But again, we see here that we have an ethyl and amine head group. Here we have serine. Here we have choline. And sphingomyelin isn't built the same way as a phospholipid. I'll show you how in a second. But when you're done, you get something that's very, very similar to phosphatidylcholine. So sphingomyelin and phosphatidylcholine, even though they belong to different classes of lipids, are actually structurally pretty similar. So let's look at how we can build sphingomyelin. So sphingolipids are found in cell membranes. So their backbone is sphingosine. So this whole structure right here is sphingosine. So this drawing from our textbook shows how sphingosine is built from a palmitate fatty acid and from serine right here. So this whole thing is called sphingosine. What we can do is we can add a fatty acid here, and instead of an ester functional group, we'll get an amide. And we can add a phosphocholine group right here because we have an OH group. So if we do that, we get a ceramide. So ceramide is sphingosine. So sphingosine right here is shown completely in black, and we have our fatty acid right here. So if we take sphingosine plus 1 fatty acid, we get ceramide, and then what we do after that is we will take one of these OH groups and add a phosphocholine group to it. So here's our phosphocholine. So again, the sphingosine is shown in black. This is an acyl group, but this is a long chain fatty acid right here, which is shown in this blue color. And here's the phosphocholine. So remember, this was an OH group. There's the phosphate group, and there's the choline. So if you take ceramide plus phosphocholine, you get sphingomyelin. And if we have the space-filling group here, you can see here we have our 2 long polar tails, but they're straight and they pack together a bit better than the phospholipids do. But you can see you've got a polar head group and 2 nonpolar tails. And again, the artist here used green instead of the CPK coloring black. But you can see you've got the polar head group and 2 nonpolar tails. So sphingomyelin is amphipathic as well. So here we can see that structurally, as I mentioned before, that sphingomyelin and so this artist calls this phosphoglyceride, but this is phosphatidylcholine, sorry. And here you can see that structurally they're very similar with their polar head groups and their 2 nonpolar tails. So again, sphingolipids and phospholipids are amphipathic, and they are found in membranes. So before we turn to membranes, let's talk about glycosphingolipids. So here we take ceramide, which is our sphingosine backbone, and 1 fatty acid, and we can add either one sugar, either glucose or galactose. And if we just have one, we call it cerebroside. But if we add quite a few monosaccharides, which are shown here, we have something called a ganglioside. Again, we still have two nonpolar tails and a fairly large polar head group, and you can tell by their names, cerebroside and ganglioside, that these kinds of glycosphingolipids are common in nervous system tissue. So let's just digress for just a second to talk about what steroids look like. So steroids have this cyclopentanoperhydrophenanthrene ring. So it's just three 6-membered rings and a 5-membered ring. So any molecule that has this kind of structure is referred to as a steroid. They are of eukaryotic origin. You don't find them in bacteria. So again, any kind of derivative, you can add different functional groups to these things. But if it has these 4 fused non-planar rings, it's classified chemically as a steroid. For example, cholesterol. So cholesterol is a steroid. So again, if we add this OH group, so this is why this is cholesterol, because we have an alcohol functional group, and we have this this aliphatic group right here. This is a major component of animal plasma membranes. You don't find cholesterol in plants, although plants do have other sterols. It's just they're not cholesterol. They're other types of sterols. And if I again went back to the same artist again, so again, if you substitute the green for the black, you can see that most of the cholesterol is non-polar, but this OH group right here gives it a little polarity. So cholesterol is also amphipathic. Here are some other representative steroids. So cortisol is a glucocorticoid that regulates metabolism, especially during stress. Testosterone is an androgen. Estradiol is an estrogen. Aldosterone is a mineralocorticoid that regulates water balance. And you can see that they all are classified as steroids because they have that planar ring system. All right, so let's go back to membranes. So amphipathic lipids can form bilayers, and they do this as a consequence of the hydrophobic effect that we talked about in class earlier, where the hydrophilic heads that are shown in this diagram in this pink color, water is happy to interact with them and forms favorable intermolecular forces with these polar head groups. But favorable intermolecular forces between water and these nonpolar tails, which are shown in green, cannot form. So that means that the nonpolar tails are forced to interact with each other, sequestered away from water. So again, this is a consequence of the hydrophobic effect, which we described as the refusal of water to interact with nonpolar domains. So back in chapter 2, we talked about micelles, where we looked at detergents that have one head and one tail. These would be conically shaped and would pack into a structure called a micelle, again as a consequence of the hydrophobic effect, where the polar head groups interact with water, but the tails interact with each other away from water. If we have phospholipids or sphingolipids, they have one head and two tails, and they're more cylindrically shaped, so they generally don't form micelles, and this is what they form. They form something called this lipid bilayer. So we get a different structure. So instead of a spherical structure right here, we get a lipid bilayer which can enclose an aqueous compartment, which the micelles do not do. So if we look at which lipids can form bilayers, hopefully this diagram shows that very well, that with these cylindrical shapes, if you have a head group and two nonpolar tails, that you form a bilayer quite nicely, whereas you have this conical shape right here that leaves you a lot of holes in your membrane. So the fatty acids on their own don't form membranes. The triacylglycerols don't either. I don't particularly like this diagram. I think that it's misleading because it shows the triacylglycerol as if it existed as a polar head group and three nonpolar tails. It doesn't. The triacylglycerols are completely nonpolar, and for that reason they don't form membranes. So the glycerol phospholipids and the fatty acids are both amphipathic molecules. The glycerol phospholipids form membranes, the fatty acids don't, and the triacylglycerols don't because they are completely nonpolar. So lipids, bilayers are biologically important. If you take our lipid bilayer and imagine that you can enclose an aqueous compartment. So this is the basis of a cell, where you have an aqueous solution in the inside of a cell and an aqueous solution on the outside, where again you've got the polar head groups on this inner leaflet of the membrane facing the aqueous solution in the inside of the cell, and the polar head groups on the extracellular leaflet interacting with the aqueous solution on the outside of the cell. This is the beginning of forming membranes. We're not there yet, because membranes consist not just of lipids but proteins too. But we can artificially make these structures called liposomes in the lab, and we can use these as a way to study how membranes behave. So if we were going to look at a membrane, so again, this is still not a membrane because we would have proteins as part of this, but I haven't added the proteins yet. But we do see in the membrane that we have the outer leaflet and the inner leaflet are not the same, that membranes are asymmetric. If you look at our key here, you can see that phosphatidylcholine and sphingomyelin are largely found in the extracellular leaflet that face the extracellular space. These hexagons here are meant to represent sugars. So the phospholipids that have sugars as part of their polar head groups, again, you see sugars on the outside, not on the inside. And the phosphatidylethylamine and phosphatidylserine are found on largely on the inside. The other thing to note is that of these phospholipids, phosphatidylserine is the only one with a negative charge. So if we go back to the structures I showed you here, the phosphatidylethylamine has a positive and a negative charge that cancel to give a phospholipid that's overall neutral. But here for the serine, we have a positive and two negatives. Again, phosphatidylcholine is overall neutral, and so is sphingomyelin. So that means our negatively charged phospholipids are located on the inner leaflet facing the cytosol, so you do have a charge difference across the membrane. We can note that here too, and we see that the phosphatidylserine is 100% located in the inner leaflet, whereas the other lipids, we can say that sphingomyelin and phosphatidylcholine are more likely to be found on the outer leaflet, and phosphatidylethylamine is more likely to be found on the inner leaflet. So I want to go back to the fluidity that I showed you at the beginning, where I showed you that the little cartoon of the membrane sort of wiggling. The lipids are highly mobile in the plane of the bilayer. So if I look at this, if we label this one in this blue color, we can see that lateral diffusion is quite rapid because the membranes are fluid. Sort of like olive oil. But this transverse diffusion, or flip-flop, without any assistance is unfavorable, because a polar head group would have to go through this whole domain of nonpolar tails, which is energetically not favorable. So the transverse diffusion, or flip-flop, is very slow. So let's add our proteins now. So this is the Singer-Nicolson fluid mosaic model. So it's been refined over the years, but it's a pretty good model, considering it was proposed about 50 years ago. So here we have the phospholipid bilayer. We have proteins that are associated with the membrane. They might be integral, in which that they completely span the bilayer. They might be transporters, in which case they would have to span the bilayer. Some membrane proteins are peripheral membrane proteins, which means they're just associated with either the outside or the inside of the cell. And again, these pinkish colors right here are meant to represent sugars. We have some proteins that could be anchored via these sugars right here. So this is a lipid anchored protein, where we have sugars right here, and this is linked to a fatty acid right here. This is another way that a protein can be associated with the membrane. So Singer and Nicolson, this was their proposed model, and the artists here drew this undulating wave to show that the membrane is a fluid sea with protein icebergs floating in it. So of course we have to, if we were going to accept the fluid mosaic model, we need to verify it experimentally. Another model that was proposed is the sandwich model, where you have your phospholipid bilayer here in a protein sandwich facing the outside of the cell and protein on the inside. But this doesn't explain how proteins would be used as transporters to ferry substances across the membrane. So the fluid mosaic model is more consistent with that view. There's freeze fracture electron microscopy, where you can actually peel these apart and look at them under the electron microscope. Cell fusion experiments, and a experiment called fluorescence photobleaching recovery. So let's look at the cell fusion experiment. So this was done at Johns Hopkins University by Michael Edidin, and he took a mouse cell and labeled the mouse proteins with fluorescent green markers, and a human cell with fluorescent red markers, because humans and mice have different proteins on their surfaces, and he induced these cells to fuse. So in and of itself, the fact that these cells can fuse indicate the fluidity of the membrane. And if you look under the microscope, you can see initially that you've got the green proteins on one half and the red proteins on the other half. But if you wait a bit, and you'd have to look these under different wavelengths to see, because they fluoresce at different wavelengths. After 40 minutes, the red and green markers have completely intermingled. So that means that at least for the ones that are able to move from one side of the cell to the other, there's nothing that's restricting their movements. So this is good indication that of the fluidity of membranes. This is another experiment called fluorescence recovery after photobleaching. So that is shown in this video. So I'm going to go ahead and go to this video right here. And
stuck looking through dozens of lessons to find help on that one topic? Or do you have a test coming up?
Skip the ad.
The lateral mobility of membrane proteins can be measured in living cells by FRAP, which stands for fluorescence recovery after photobleaching. For this purpose, membrane proteins are often expressed as fusion proteins with the green fluorescent protein GFP and observed with a fluorescence microscope. A selected area of the cell is then bleached with a strong computer-controlled beam of laser light. Those membrane proteins that are not anchored and therefore can diffuse in the plane of the membrane quickly exchange places with their neighbors and fill back in the bleached area. From the rate of this fluorescence recovery, the diffusion constant of the protein can be calculated. Here, GFP is fused to a membrane protein that lies in the membrane network of the endoplasmic reticulum. After bleaching, we observe quick recovery of the fluorescence, showing that the protein is very mobile in the plane of the membrane. The same experiment can be repeated using a protein that is firmly anchored and not free to diffuse. Here, we observe GFP fused to a protein of the internuclear membrane that binds tightly to the meshwork of the nuclear lamina. After photobleaching, no fluorescence recovery can be seen over the same time frame.
All right, so let's go back to our PowerPoint. So when you bleach this area, any proteins that are free to move will move into this area because of the fluidity of the membrane, and you can actually over time measure how long this takes. But as the video shows, some proteins actually are anchored, and if they're not free to move, then you won't see the recovery. So the other thing that we can notice is, and this makes sense in terms of the fatty acyl chains that you have, that membrane fluidity is temperature dependent. And it does depend not just on temperature, but the length and the degree of saturation of the fatty acyl chains. So here we're above the transition temperature. So above the transition temperature, a membrane will be very fluid. So these fatty acyl chains are free to move. But if you go below the temperature, the transition temperature, then this slows down the movement, and now the fatty acyl chains are able to pack together, and they interact via the London dispersion forces. So they form more of a gel type structure. So at exactly what temperature this happens is going to depend on the fatty acids, how long are they, and do they have lots of double bonds in them or not, because this goes back to what we said about the beginning about how the saturated fatty acids pack together really well, whereas if you have the double bonds, they have lots of bends in them and they don't pack together as well. So I have another membrane, a video showing membrane fluidity. So let me show you that.
I was never able to find a bra that fits my small chest. For as long as I can remember, being small was something to be.
That's unfortunate. Okay. Inappropriate.
A piece of the plasma membrane of this neuronal cell is pulled out with laser tweezers. Remarkably, moving this membrane tubule rapidly back and forth does not rupture the plasma membrane, which flows quickly to adapt to the mechanical distortion.
Anyway, I thought that you might be interested in that because you can observe the fluidity directly in that video. So the other thing that we should notice if we come back to our steroids, remember I showed you the cholesterol was also amphipathic. So the transition temperature depends not just on how long or how many double bonds the fatty acids have. It also depends on the cholesterol that's part of the membrane. So here I'm going to go back to another video and hopefully they won't make us watch an ad, but they
the right care at the right time.
Here we go.
Plus 20% off
Just wait a few seconds.
Welcome back your smile today.
The head groups and the phospholipids.
Actually, it looks like I was right in the middle of watching this.
Phospholipids contain a head group, choline in this case, that is attached via phosphate group to a 3-carbon glycerol backbone. 2 fatty acid tails are attached to the remaining 2 carbons of the glycerol.
So there's that bend in the unsaturated fatty acid.
The head groups and the phosphate are polar, that is, they prefer to be in an aqueous environment.
So that's our polar head group.
In contrast, the fatty acid tails are hydrophobic, that is, they are repelled from water. The fatty acid tails on phospholipids can be saturated with no double bonds, or unsaturated with 1 or more double bonds. The double bonds are usually in the cis configuration, which introduces sharp kinks. When forming a bilayer, unsaturated fatty acid tails pack loosely, which allows the bilayer to remain fluid. If there were no double bonds, bilayers would solidify to a consistency resembling bacon grease. Cholesterol is another lipid component of most cell membranes. It has a hydroxyl group, a tiny polar head group, so to speak, attached to a rigid hydrophobic tail. Cholesterol can fill gaps between phospholipids and thus stabilizes the bilayer. In a lipid bilayer, lipids arrange themselves so that their polar head groups are exposed to water, and their hydrophobic tails are sandwiched in the middle.
All right, so I thought you might enjoy watching that. Okay, so that is the end of our lecture. So if you're watching this during the week that we go through this lecture, we are going to do a POGO activity on the flippase enzyme, and that enzyme will describe how the membrane asymmetry that I showed you is maintained. So thank you for your attention, and I will see you in class.